why the heck are we doing DNA sequencing? More than anything, it falls under that big umbrella of conservation. If you're interested in conservation, one of the most important things is you have to know what you're trying to conserve, which means you have to come up with a good accounting list of species that are out there. This is a big goal of the DNA sequencing work that we do. We just want to come up with a proper accounting of what is out there and whenever I say a proper accounting, I'm talking about research grade observations that follow a specific protocol and a repeatable protocol. So that's what we've been trying to do a couple years. And I feel like the work has come a long way, and we continue to pick up speed. We had a very good year in 2023 by my own estimation, and we have ambitious goals set for 2024 again. We'll meet them all. Set the goals. Set the goal beyond your reach. So what we do is basically every month of the year we meet and we have a lab day. Right now we meet at the Mount Rainier Nature Center, and what we'll do is we use mini PCR, that's polymerase chain reaction, to process a small sample, that's 16 samples batch, do that on a weekend and a day. This is slow, detail-oriented work. We'll go there at o'clock. Well, we usually schedule it till 4, but I don't think we're there by 4 p.m. We're still cleaning stuff up till after 4. So, it's time. It's January 24th. And so if that's something, and I would encourage people to come out and join us and ask questions and observe us on a lab day, or potentially if you want to come up with mushrooms that you want us to run sequencing on, this is a capability that is open to club members, but you have to follow a certain protocol in order to give us a mushroom that we're going to do DNA sequencing on. So the best thing, if you're interested in this, is come to a lab day and ask us questions, and we can share that knowledge base with you. Also, the lab days, as soon as we schedule them, generally, I post them on the front page of the website, so you can stay current with upcoming lab days if you check there. As I said, our base of operations for doing lab days is at the Mount Rainier Nature Center. But we also picked up another sample and lab day location this year. This is at the Jug Bay. There's a picture of Jug Bay there. That's a really beautiful sanctuary. We'll go down there a couple times in the coming year as well. And another place that I want to pick up for occasional sampling is the Clifton Institute over in Virginia. I went out there to give a talk on fungi and led a walk, I don't know, a couple months ago. And there's a photo from that event where I'm showing this young future mycologist a glorious giant puffball. So we'll go back there a couple more times in 2024 to sample and add some data points to the Clifton Institute. And we're looking for other places to partner with as well. That's because, another success from 2023 is we were able to bring Stephen Russell in as one of our guest mycologists at Sequanota. Sequanota is our weekend event in South Central Pennsylvania in the Laurel Highlands there. And Stephen Russell is the founding person of the Hoosier Mushroom Society, and he's probably run more DNA sequencing than, I don't know, most anybody, I imagine. And he just has so much experience with this stuff. He gave what I felt was a really great talk at Sequanota, and then he and John Plischke they collected approximately 690 samples there, which is just an absurdly large amount of samples, which Stephen took back with him to his home, and he's going to run those through. He's going to do that sequencing work for us. So that's going to take that 690. That really moves the ball down the field for us, because when it comes to this DNA sequencing work, it's really all about more data. You just need more data points, because eventually, as you go out and you sample more and more and more, the curve will flatten. After you get enough data points, you're not going to be discovering nearly as many new types of fungi, and that's the goal here. It's really all about doing more and more samples, statistics. We have some goals for 2024. As I said, our next lab day is going to be at Mount Rainier on January 24th. I'd encourage people to come out and bring some questions with him, and feel free to stop by. Like I said, I'd encourage that. Also, I kind of have been splitting my time between Central Pennsylvania, where I help out with aging parents, and DC over the last couple years. But in 2024, I'm going to be in DC a lot more. So I'm planning on hosting DNA sampling walks probably once or twice a month, especially during months where the microfungi are cooperating, and these walks, just be warned, they're not like regular walks that you go on with Mitch. Everything about sampling for DNA, you have to slow everything all the way down. You have to take good photos, take good notes, etc. But we're going to be doing a lot more of that in 2024. I also signed up for a potential nanopore training session with Stephen Russell. Nanopore is the next technology. This allows you to process a lot of volume in a hurry. And we also want to develop some closer ties with Fundus. This is the branch of NAMA, the Fungal Diversity Survey, which is interested in promoting this type of DNA sequencing work. We want to develop better ties with them. And again, just make another call to you. This is very important work, and we could use your help. So if this is something that appeals to you, we'd love to see you come out and join us. And then with that, I am going to finally stop sharing and shut myself up so we can listen to Megan the rest of the way out. And again, thank you.
Megan, actually, before you jump in, Saranella has her hand up, so I'll let her chime in a little bit too, because I know Saranella has been very involved in this.
Yeah, just thank you, Elizabeth, and thank you, Tom. I just wanted to do a couple of additional information. If you are coming to any of our lab days and you are coming to ask questions and to establish a conversation with us, the best time to arrive is 1 p.m. when the PCR machine is already running. Now, if you are planning to bring russulas or boletes for us to sequence, then please arrive by 10 a.m. so we can include those samples in the batch for the day. I also wanted to make a small correction to the date. Our next lab day, it's going to be Sunday, January 21st. And the second one will be Saturday, February 24th. I saw Tom combine the two dates there. So lab days are going to be January 21st, the Sunday, come at 10 to give samples or come at 1 to talk. Thank you.
Thank you, Saranella. And I'll add to that if you do want to bring, and we'll talk in a second about why russulas and boletes, but if you do want to bring something to sample, it would also be really helpful if you let us know ahead of time. As Tom mentioned, we get there at about 10 in the morning and we start going through the process. It's not something that's kind of rolling. You started at 10, you finish around 4:30 with the batch of 16. And so if we are waiting, if we start at 10 and you don't show up till 11, we can't incorporate that. But if we know that you're going to come we can make sure to set aside a spot for something you might bring. So just be in touch if you have something that you want to bring by, and that would be helpful. All right. So you can all see my screen, right?
Yep, looks good.
All right. Yep. All right. So in 2023, we obtained about 70 good sequences, which is about 1/10 of the sequences that Stephen Russell is going to be getting us from Sequenota. And part of that is because, as Tom mentioned, we're doing small batches. We're focusing on the small batches of specific things, partly because these days are not just about generating the sequences. They're also about providing a space where people from the community can come and see what we're doing. And so last weekend, for example, we had 2 high school students and a teacher that came and saw what we were doing at the end and asked a lot of really good questions. We had 2 people that were apparently visiting from Texas and saw that we were having a DNA day and thought that would be how they wanted to spend an afternoon in the DC region. So that's maybe not what I would have done, but that's really exciting that people want to come see what we're doing. So we got about 70 good sequences this past year, 40 different taxa. And part of what we were doing the first 2 years was sort of proof of concept. Can we get good sequences out of things that we are bringing in our process? And we can, and we're happy about that. So for 2024, we wanted to focus a little bit more on some specific groups. We don't really need to keep on sequencing another Trametes or another Trichaptum biforme or another of things that we've done a lot of. And I'll mention in a minute why Russula, but we've decided to focus on Russula and Bolitacea. That doesn't mean that if you have something interesting that you say, you know, I really think that we could get a DNA sequence out of this and answer some questions, we aren't going to say no, but we just really wanted to kind of focus our efforts. Another thing we'll be doing this year is adding our sequences to GenBank, which I'll talk about in a minute. Currently, all of our sequences are on INAT as a text sequence, but we will be adding them to GenBank, and we will be analyzing the sequences from the Sequenota foray once we've gotten them back from Stephen Russell. So I mentioned our iNaturalist group or our iNaturalist project. Anyone can go and look at that. It's the MaDNA iNaturalist project. Currently, we have 176 observations. You will be able to go and see the observation and see the text of a DNA sequence if we were able to obtain one. So when we talk about DNA sequencing, I think a lot of times people will think about a lab and some setup like this. And this is not what we're doing. We have a set of 4 boxes that contain the equipment that we need to extract DNA and to do the PCR. And then we send it off for commercial sequencing. And here is the space in the Mount Rainier Nature Center where we do our DNA extraction and set up the PCR. So we get a little bit of fungal tissue and put it in a tube. We add some chemicals that help break open the cells and get out the DNA and separate the DNA from proteins and sugars. Oops. So this would be a Russula that maybe we'd gotten some tissue out of. We then run the PCR from a little bit of the DNA that we've extracted. We use gel electrophoresis to visualize the product and see whether our reaction worked. And what we would see then is this sort of thing, which is a gel. On the left-hand side, you have a size ladder. That's a standard thing that you put in there, and it tells you whether what you've got is 250 base pairs long, or 500, or 1,000, or 1,500. And these bands here, these lines, tell us that the DNA PCR, the polymerase chain reaction, which is a way of making lots and lots of copies of DNA, these tell us that it worked. So in this case, we had a number of samples that worked and a few that didn't. These columns that don't have anything in them would be ones that didn't work. So we put them in this machine, which runs through a lot of temperature changes to make copies of the DNA. We use this to visualize the gel. And then these that we have a positive reaction for, where we amplified DNA, we would send these off for sequencing. And what we get back from the commercial company, GeneWiz, is something that looks like this. This is a chromatogram, and it gives us our DNA sequence. You see all these little peaks that correspond to nucleotides, and they're all color-coded, which is kind of nice. And then we can take that DNA sequence and put it into a computer program and use that to analyze our sequence. And one of the things that we do is we compare it to other sequences in a global database of DNA sequences. So the NIH has the National Center for Biotechnology Information, and that is a globally used database of DNA sequences. And you can use this tool, a basic local alignment search tool, to compare your sequence to all the other sequences out there. And these are sequences of fungi, of plants, of animals, of other things. We, restricted to fungi, but sequences of everything are out there. And what you get back when you do that blast is something that looks like this. So you get sort of a list of things that are similar to your sequence. And in this case, they are ordered by certain parameters. And the ones to look at here are the query cover. So you put in your sequence and it compares it to all the sequences in the database. And it shows you that across the length of your sequence, it was 100% compared to this sequence. And the percent identity is the other thing to look at. So they completely aligned. You had two sequences that were about the same length, and they were exactly the same along that length. And that gives you some good data that what you have is the same as what someone else put into the database. And so in this case, it's a Russula that Stephen Russell put in, and you can go back to this iNaturalist entry and look at it. Another thing to notice is the percent identity for these are 100, drops down to 96, and then it drops down pretty quickly. So what that tells me is that in this database, there are only about 5 things that are very similar. When you drop down to 96 and below, you're dropping down to a different species at least. And so of all of the sequences in the database, only about 5 have been put in of this particular species. So there aren't a lot of entries in the database of this particular species, which matches to one of the ones that we've been looking at. So you can get information about, is this something that's super common? Is it widespread? Is it closely related to a lot of other things? And that's what we're using this information for. Now, as I've been talking, maybe you've been kind of reading through this and you see Russula spa, uncultured ectomycorrhiza, Russula spa. And the second one here says Trichlomopsis decora. Well, anybody can put anything into this database. It's kind of like Wikipedia. And so you can put anything in, you can put any sequence in, and you can call it whatever you want. And so sometimes there are mistakes. Sometimes there are things that you have to kind of pay attention to and say, well, maybe they switched up their samples. Maybe they mislabeled. Maybe they made a mistake putting it in the database. We don't know. But it doesn't necessarily mean that Russulas and Trichlomopsis are all of a sudden the same thing or super closely related. It's probably something like this means it's probably a mistake in the database because Trichlomopsis, I've got a picture further down. Looks nothing like a Russula. All right. So, although we're focusing on Russulas and boletes, we take in other things that people are interested in, and we'll have those entries in our iNaturalist project. So this is one that Martin Livesey put in, and he had kind of guessed at a name and brought it in for us for sequencing. So this is what you would see in the iNaturalist project. And then further down from that, you've got, your tentative IDs. And then you've got the ID that I put in based on the DNA sequencing. And over here in the observation fields, you've got our DNA barcode ITS sequence. So this is what a complete entry in iNaturalist should look like for anything that we have sequenced successfully. We will have the ITS sequence that anybody can go and copy and put through the BLAST search and align with other DNA sequences. And we'll have a suggestion based on what that BLAST top hit was. So that very first one on the list, people tend to call a top hit. And then you've got the other hits further down. So in this case, the BLAST top hit with 100% query cover and a 99.86% identity was this GenBank obsession, which was Gymnopus foetidus, and other iNaturalist that you can go look at. So another one that we were able to sort of refine was this. When this was initially put into iNat, the tentative ID was order Polyporales, which is right on. Here's a close-up of what we were looking at there. And after doing the DNA sequencing, the closest match was Björkendera adusta. So that's just an example of some of the things that you can do. Sometimes you have a very small amount of tissue that's not very descriptive, and you can use DNA sequencing to get a much better ID. This one was interesting. It's a Pseudotrichoma ambrosum. You know, the initial ID was Kingdom fungi. I'm not really sure what this is. We were able to successfully get DNA out of it. The top hit was the type of Pseudotrichoma ambrosum. Here's a close-up. It's a very nice one. And that is a mushroom that actually does not have that many entries in iNaturalist. So they seem to all be from the Northeast, starting down to the mid-Atlantic. There's this one down here in Tennessee, but it's a taxon that's really not widespread in iNaturalist and in other records. So that's pretty neat that we have a really good, solid record of it based on the DNA sequencing. And here's another one, initial entry, something in the Basidiomycota. Annie guessed maybe Cortinarius, and Annie was right. Annie's probably always right, so just believe her if she says something. Because after we did the DNA sequencing, that was definitely a Cortinarius. It might, I think it's a little wet there, so it may not look like sort of a typical Cortinarius, and that's the only picture that I sort of expanded. There are some other pictures of the gills and the stipe that you can go and look at. And this was another one where we were able to really kind of refine the ID. Family Marasmiaceae ended up being a Calabiopsis biforme once we did the DNA sequencing. So we've been able to answer some questions of some of these little mushrooms that maybe if we'd spent a lot of time, maybe we would have gotten there. We were able to get there pretty quickly with the DNA sequencing. But all of those are in our project and have the DNA data there. And we save each of our samples. So we make sure that they're dried down and they're saved so that someone could go back to them in the future if they wanted to use them as part of a project or something else. So like I said, we got about 70 good sequences, 40 different taxa, but we wanted to focus really on we wanted to sort of grab onto some things and chew on them a little bit more. And so we're going to focus on Russula and Boletaceae. And why Russula? Well, it's a really big genus. Of course, you can eat some of them. It's not one of the choice edibles. Some of them will make you sick. You can't use it for dyeing, so I don't really care. But it's a really pretty and widespread genus, and one that's really hard to ID based on morphological characters. And I really liked in iNaturalist, actually, they have this quote: if we know of anyone who in the pride of intellect spurned all mental tasks as mere play, we would tame him by insisting on his mastering, classifying, and explaining the synonyms of the genus Russula. And if you've ever gone to Michael Kuo's The Mushroom Experts page on Russula, and I recommend that you do, he has a great diatribe about Russula and how hard they are to identify. You, in a lot of cases, need to use microscopy, and even microscopy, because of the variability of Russulas, won't necessarily get you to species. But precisely because of how difficult they are to identify based on morphological characters, we thought they're a really good one for DNA sequencing. And there is some information out there. People have been putting together monographs and other papers on russulas. So there is some information out there, some sequences we can compare things to and get some more information about what we're seeing in the area. So this is an example of one put in as genus Russula. And this is the top hits, as I showed before. What this does suggest is that this is one that's just not widely documented. We can discount this Trichlomopsis as something. There's an uncultured Exomycorrhiza, and then there's this other Russula spa and this other Russula spa. So this is probably an unnamed species. Stephen Russell puts on these sort of monikers so that he can tell one unnamed species from another. But the fact that it's the same as all of these, but that these are this much different from other ones and don't have a name means that this probably is an unnamed species. This is ours. This is one of the ones that Stephen Russell has sequenced, and this is another one. So you can look at them and they are very similar. They are just another white Russula. But now we have information that the ones that ours was closest to, the DNA sequences of ours were closest to these others. This is where these white Russulas can be found. So now we have documentation that we have one here and that they have been found in these other places. And so you might go into the woods and you just see this white Russula, say, oh, it's Russula spa. It's a white Russula. Who knows how widespread it is and who knows how much different it is from other ones. But this gives us good information that, well, it might not be all that widespread, or it might not be all that widely documented because it looks like other things. And now we've got some solid information about where you can find this particular species, even if it doesn't have a name. Whatever this sequence represents something that can be found in these different states. Sort of an opposite scenario is with Russula cyanoxantha. This is an observation from Tom McCoy, I think. And it's a sad little thing, but we were still able to get some pretty good DNA out of it. And it came back as 99% query cover, 99.8% identity to Russula cyanoxantha. And here's another entry in iNaturalist for that species. And this was a more recent one that we also got in. And what was really interesting is this one was collected several months later in a completely different location, and they were identical in terms of their DNA sequences. And when you look at what's in GenBank for Russula cyanoxantha, you can see that they are all nearly exactly the same. And what that says to me is there's either sort of a population that's very, very similar around all of these places that it's been collected, or the ITS region doesn't resolve species for this group. Either one could be happening. If they are all sort of a population of the same thing, then that's really interesting because you're finding it from Wisconsin to Tennessee to Panama to Nova Scotia and throughout the US. Russula cyanoxantha is one of the Russulas that they claim is easier to identify than other Russulas because the gills are not as brittle, they're more pliable, but it shows a wide range of coloration here. These two are sort of tan, it can be purplish, it can be reddish. They all seem to be sort of mottled though. And all the pictures, if you look through an iNat, a lot of the pictures, something's been chewing on it. So obviously it's not just, it's considered edible for humans, but I think other things must like it too, because there always seems to be some sort of slug damage or squirrel damage or something that's been chewing on it. So again, now we've got good documentation that this Russula that looks the same, and the question is always, well, is it really the same? In this case, it seems like it really is, and it's really widespread. So another Russula that we worked on was this one that was collected in the Sequinotophore. When we did the blast search, this was our hit table You have a few at the top that are 99, and then it drops precipitously to 97 and then on down. In this case, again, there's no good name. It's Russula spa. It may be an unnamed species. A lot of the entries are also uncultured ectomycorrhizal, so they're soil samples or root tip samples. And that's the other thing that we can, get out of this information is these are fruiting bodies that people have collected and sequenced, but there's also a lot of soil studies that are going on. And this just gives us more information about how widespread these species are, even if they don't have names. And so in this case, this is one from Sequanota. These are two others from our DNA project. And then these are two from iNaturalist that are there on our DNA hit table. And another thing to note is the color variation. These all look a little bit different. These are more close. You've got that peach colored cap. These have sort of a white colored cap. Is that a true variation? Does that mean two different things? Or do you just have sort of color variation in this Russula that is all very similar based on the DNA sequencing? So every time we answer a question, we end up with about 10 more questions to ask. So like I said, a number of the hits that are on that hit table are from fruiting bodies. So there's that Russula is in Indiana, it's in Pennsylvania, it's in New York. And then based on soil sequencing, it's also in Michigan, North Carolina, and Massachusetts. And so if you were only getting a root tip or only getting a fruiting body and looking at the morphology, and even with an iNaturalist observation, it would be hard to say that these are all probably the same thing. But looking at the DNA, you can compare across these different entries and say, okay, this Russula, even if we can't put a name on it, occurs in all of these different places. So this is some of our Russula results from 2023. We actually got in another batch of about as many this morning, and I haven't gone through them yet. So we've got hot off the press information as well. So like I said, we go through, we do the blast ID, get a top hit, put all of that information in iNat, and this year we'll be putting the sequences in GenBank so that other people can compare what they're seeing as well. We haven't put as many in GenBank yet because to put them in GenBank, you have to annotate them, you have to answer a number of questions, and it's a little bit tedious. And I just haven't wanted to do it one off at a time. But we will be putting those in. And another thing that you can do with the sequences is in various software programs, align them all. And then you can kind of generate this relationship tree to see where yours land in context with other Russula species. So here I downloaded a number of sequences of different types of Russula. So type is the specimen that was used to describe a species. So when you describe a species, you say, this is the type specimen, and this is sort of the gold standard. This is what I'm going to base everything on. If you have a sequence from that, it's an X-type sequence, and there's a reference database in GenBank that you can look at for that. So I aligned those sequences and looked at ours compared to those. And so the ones with the red arrow are where ours land. So we've got a pretty good diversity even so far with just the few Russulas that we've collected, but I'm looking forward to having various collections that cover some of the rest of the diversity that might be out there. All right. So I'm going to stop sharing with that and ask if there are any questions.
Thanks, Megan. And I'm going to say we're down to few enough people that I'm just going to say if you have questions, feel free to just jump in. You talked a little bit about this, but Elizabeth actually asked how you can be sure that the databank top hit is actually the species that it says it is.
That's, well, yes. So that's sort of the reliance upon the integrity of the scientific community. And so if you have something in there, it should have a voucher number ideally it goes back to some sample somewhere. It should be well documented. Often you'll look, is it part of a publication? Is it something that you can go back to? So you're relying on the person that put it in, having done a good job of keeping a voucher specimen, doing a good job annotating their sequence, it's built on trust, that community. And so you can, like when we compared there and we saw the one that said Tricholomopsis, you can sort of go through and take sequences and compare them back again and see where they land and, go, okay, well, they called it a Tricholomopsis, but it's obviously not, and sort of discount that sequence. But
is there
using-
any way to like contact the person that labeled it and change it?
Yeah, you can. There's really not an editing function. I have at times contacted the folks at NCBI and said, there's a typo here, or I know that this goes back to this specimen. Can you contact the original people and ask them to change it? But yeah, I mean, there are a lot of issues in GenBank. People will publish a genus and a provisional species, knowing that they're going to change the name, but after they publish the paper, they don't go back to GenBank and annotate and update the name. So you really have to be careful when you're looking at it. You have to kind of look at each entry and follow through on it and look for the publications and look for who put it in and really do your homework. So you can't just take that top hit and go, well, it says the top hit says this, so it must be that. You need to really sort of analyze what you're seeing. Okay, did that answer the question? I hope so.
I was thinking as you were talking, because it really hadn't occurred to me that this connection, but when I was working on Undergrove, which is about the mycorrhizal relationships between specifically Douglas fir and the mushrooms that it is connected to, I went and looked up a bunch of scientific papers of like, what do they think is mycorrhizally connected? And it is these, soil sample or, digging up some roots and washing them off and figuring out what mycorrhizae are in there, and they DNA sequence them. And they don't know what they are, right? Unless they find a hit in the database. So there's a lot in those papers. There's always a lot of rows of like, just numbers of like, we didn't know what this one was. We didn't know what this one was. And so that's fun to think about how our little mushroom club could be like filling in some of those
Right.
blanks for people.
Yeah. Yeah, there's this, fungi is this vast kingdom that has been hardly explored.
Yeah. Does anyone else have questions or comments? You want to jump in? It's getting late. All right.
Seems like it. Well, yeah, like Serenella said, folks are welcome to come stop by in the morning if you want to drop something off. We do tend to, the very first part of the process, we sort of need to focus and kind of don't talk a whole lot, or shouldn't talk a whole lot. So when we get the samples in, we're doing the DNA extraction, trying to put numbers together with other numbers and line things up. Usually around 1 o'clock, we're running the gel. And at that point, we have time to sort of talk about what we've done and what it all looks like. So, but we love having people come by.
Very cool. Well, thank you, everybody. And thanks to Tom for all your work tonight, too. All right. Have a good night.
Night, everyone.
Bye-bye.